So let me start a little bit with something about myself. My education is in cancer research. I came to this country about 25 years ago from Brazil. Exactly, I came from warm Rio de Janeiro in February with 40 Celsius in Rio de Janeiro to Boston, below 30 Fahrenheit. Was a cultural shock, but also thank you to that scholarship that brought me to this country to develop my doctoral studies. It kind of changed my professional and also my personal life, as I've raised three kids in this country. Throughout my career, I had opportunity to work both in academia and industry. And if you ask me what made like connect the dots of my career, is like a big scientific question of how to use genomic data to answer questions that will help diagnosis, treatments, and prevention tools for doctors, patients, and families. So the Kids First could not be a better fit for me in my background. Data resources that I have worked in the past, usually, more often than not, they are targeting one type of data, let's say genomic data, transcriptome, or one type of disease like breast cancer, lung cancer. But the Kids First program, what I learned is that by building a resource that allows multiple data types and also data from across multiple pediatric conditions, it allows a deeper understanding of the biology behind the conditions and also a richer data for researchers to work with. Also, I learned about the commitment of the program to share genomic data associated with phenotypic and clinical data, which again give additional meaning to the genomic data. I was amazed to learn about the history of the Kids First program. All started with Gabriella Miller, who died with 10 years old of brain cancer, and she was a fearless advocate for childhood cancer. And the Kids First program is part of her legacy that she left behind. In 2014, a bipartisan bill approved Gabriella Miller Kids First Research Act, authorizing $12.6 million per year for 10 years to support pediatric research at the NIH. And today, I'm happy to give you new developments. Just about a month ago in July, House passed the Kids First 2.0. We know still there are multiple steps for it to be signed into law, but nevertheless, I want to share with you this exciting news about the future of the Gabriella Miller program. The Kids First program starts with a basic hypothesis that there is an intersection between human development and childhood cancer. And through the many years of the program, our investigators, along with many researchers around the globe, are generating data substantiating that hypothesis. For instance, I want to point out, highlight validated clinical targets for oncology like BRAF, MAPK, and ALK mutations that are shared also with structural birth defects. More recently, a large study involved 10 million births associate birth defects with higher risk of developing cancer in the childhood as well as adulthood. At NIH, I work with a group of colleagues that are from multidisciplinary team from across the NIH with the leadership of the Common Fund and many other institutes forming a working body that oversees the program. At NIH, we call it a working group. So as part of the working group, I'm really excited to be part of this team to build a cloud-based genomic data resource to accelerate collaborative research, leading to better prevention, diagnosis, and treatment for patients and families affected by structural birth defects and pediatric cancer. What I learned is that Kids First program is empowered by the NIH EXO-1 mechanism, the Kids First Sequencing Centers, and Kids First Data Resources Center. But always starts with the patient cohorts and your proposals. So proposals come to the EXO-1 mechanism, and I will explain a little bit more about that. Go through the NIH peer-reviewed system, and selected projects are then giving support, is an in-kind support for access to generation of data. So the NIH, through the EXO-1 mechanism, actively invest in data generation. And the EXO-1 investigator received that in-kind access to genomic sequencing that happens at the Kids First Sequencing Centers, the Brody Institute, and Hudson Alpha. Once the sequence is done, the Data Resource Center compiled the genomic data with phenotypes and clinical data that investigators promised us in the beginning. And then the NIH approves the delivery of that data back to the EXO-1 investigators, who now have 6 months exclusivity to work with the data. After that 6 months, the data is released to the public through dbGaP and the Kids First portal. So this is a cycle that can also very often, and we have seen through the years, many of these projects generate hypotheses for new projects. In this way, the NIH fostering data generation, data sharing, and data analysis. But I will tell the data analysis is coming later, and I will tell you more about it, because the EXO-1 mechanism supports the data generation part. So I want to tell you a little bit more about the data delivery, which is an important step in this cycle, this data cycle, if you will. Before delivery back, we go through an interactive and collaborative effort with our EXO-1 investigators to compile the genomic data with these phenotypes and clinical data. So it's a very collaborative process that we call clinical data review, and then it goes through a quality control before the NIH approve the delivery back to investigators. And as you can see in this graph, it's showing you that how projects being delivered to investigators since 2015. And as we won, most is blue, and some of the 2020 and 2021 are starting being delivered now. After 6 months exclusivity, the data then is released to the public for public access through dbGaP and the Kids First portal. And I would like to invite you all to register and look at the data in the portal. We already have genomes for almost 30,000 patients and families in our portal. And the good news is, even if you're not an EXO-1 investigator, you can have access to this data. This data is available for our NIH investigators, as well as wide community of researchers, both in the US and internationally. So again, I invite you to check it out, our data at the Kids First portal. The Kids First portal is powered by platforms such as Cavatica, and you're gonna listen more about it later today when you hear from the Data Resource Center. I wanna share with you recent data release. This is brand new. It's actually the first time the public sees this data releases that was from August to September, both in structural birth defects 2 new studies that were released in the Kids First portal and now available for researchers to work with. And again, this is not only genomic, but also some transcriptome data that are associated with phenotypes and clinical data. And then also a new release on childhood cancers. So I invite you all to check the Kids First portal and look at the new data available. We expected to release multiple data sets in the next few months, both structural birth defects projects and new cancer projects as well, adding another 5,000 genomes representing patients and families. And I want to emphasize this is available not only for EXO-1 investigators, but also for the wide research community in the US and internationally. And this gives you an idea of the data access to the data we already have at the Kids First portal and through dbGaP. As you can see, since this data was released, not only the EXO-1 investigators access it, but it's increasing access through the time of all our data sets. Most of the data sets had also an increase from May to September. We can already see a significant increase in data access requests approved by the Kids First Data Access Committee. So we have a total of 56 projects, sequencing projects that the Kids First program funded through the sequencing centers, 30 publications, 27 studies available at dbGaP and the Kids First portal, with over 500 investigators already with access to this data. Are you going to be next? The Kids First program is not alone. We are part of a bigger ecosystem, data ecosystem that exists at NIH and outside NIH. I want to mention leadership of the Common Fund and Common Fund Data Ecosystem, our partnership with Include, and you're gonna hear from today from our investigators working with Down syndrome, and also I wanna mention our partnership with the NIH cloud-based platforms, interoperability, and CPI partners. We wanna expand these partnerships, not only to create additional use cases and interoperability case, but also to start looking to functional studies on genetic variants that is being identified using the Kids First data. So our focus in 2023 is add additional data types. So genomics, proteomics, long read, diversity in cohorts. And you will hear in the near future some results from our proteomics and long read pilots. Enhance the data sharing and creating new collaborations. Again, collaborations to generate data, to analyze the data, to interoperate, name it. All are welcome. The more, the merrier. I want to finish the talk giving you a little bit of information about the EXO-1 mechanism. So what to expect? Keep in mind, first of all, read and follow the guidance of program announcement. And the EXO-1 mechanism, check it out, the past opportunities, and the URL, it's here, will be available to you in this deck. But we also welcome that you reach out to us. So feel free to email me and I will direct you to our NIH staff that can answer your questions before you submit an application. So we expect to have new opportunities for fiscal year 2023. But as I said, look at the prior announcements so you can start preparing your proposals and reach out to us to discuss it if you have questions. Now, talking about data analysis, I want to talk a little bit, as I said, the EXO-1 mechanism, the main purpose is to generate the data. But then NIH also has support mechanisms, funding mechanisms for the data analysis. And I want to highlight the RO3 mechanism, the Kids First RO3, but as well as other institutions' RO3s, which can be used to analyze the data, not only analyze the EXO-1 data, but also even if you have your own data set and one related to the data in the Kids First portal, that is also, we would welcome that type of applications. We also welcome additional adding phenotypes or longitudinal studies. So these are examples of proposals you can submit as a RO3 that will help you to analyze the data. And again, you don't need to be an EXO-1 investigator. As I said, these opportunities, both NIH EXO-1 and RO3s, are available to investigators in the US and internationally. As far as you're using Kids First data, we welcome you to be part of this community and collaborate. So I want to leave you with the Kids First vision, and I will welcome for questions. Thank you.